A pleasant day to everyone watching this video report. For today, our group will be discussing steroidal hormone drugs, their uses, and their structure-activity relationship. So, what are steroidal hormone drugs? Steroidal hormone drugs are synthetic versions of hormones that are naturally produced in the body, such as corticosteroids, anabolic steroids, and sex hormones like estrogen and testosterone. They are widely used in medical treatments for various conditions, including inflammation, hormone imbalances, and certain types of cancer. These drugs mimic the effects of natural hormones to regulate bodily functions or promote specific physiological responses. For instance, corticosteroids like prednisone are commonly used to suppress inflammation in autoimmune diseases, and this will be discussed also later on. While anabolic steroids are used to promote muscle growth in conditions like muscle wasting disorders. However, despite their therapeutic benefits, misuse or long-term use can lead to significant side effects, including osteoporosis, liver damage, cardiovascular issues, and endocrine dysfunctions. So now let's move on to the different types of steroidal hormone drugs. So first we have here is flutamide. It is an anti-androgen drug primarily used in the treatment of prostate cancer. It works by inhibiting the action of male hormones or androgens like testosterone at the androgen receptor. By blocking these hormones, flutamide helps to slow the growth of androgen-dependent prostate cancer cells. Additionally, it is sometimes used off-label for conditions such as hirsutism or excessive hair growth in women, and is part of hormone therapy for transgender women. So here we can see the structure of flutamide. So now let us discuss its SAR or structure-activity relationship. So the SAR of flutamide is crucial in determining its function. Its core structure consists of phenyl group attached to a thiazole ring, which is essential for binding to the androgen receptor. The aromatic ring, particularly the para-substitute phenyl group, plays a key role in receptor affinity, ensuring that the drug effectively competes with androgen. The thiazole ring enhances metabolic stability and strengthens interaction with the receptor, making flutamide more effective. Additionally, the aminomethyl group contributes to the optimal receptor binding and activity. These structural modifications make flutamide a potent anti-androgen drug with selective action against prostate cancer cells. So next, we will be discussing norethindrone. Norethindrone is a synthetic progestin used in oral contraceptives and hormonal replacement therapy. It mimics the action of the natural hormone progesterone, helping to regulate menstrual cycles and prevent ovulation. By altering the uterine lining and thickening cervical mucus, it effectively prevents pregnancy. Aside from its contraceptive use, norethindrone is also prescribed for conditions such as endometriosis, abnormal uterine bleeding, and menopause-related hormone therapy. Its ability to provide stable progesterone levels makes it an important medication for women's reproductive health. The SAR of norethindrone reveals key modifications that enhance its progestational activity. It has a steroidal backbone derived from testosterone with critical alterations that improve its function. The ethyl group at C17 increases progestin activity while minimizing estrogenic effects, making it more selective for progesterone receptors. The double bond at C1 to C2 enhances the drug's potency and bioavailability, ensuring better absorption and effectiveness. Additionally, the C19 methyl group plays a vital role in maintaining selectivity for the progesterone receptor while reducing unwanted androgenic effects. These structural characteristics contribute to norethindrone's efficacy as an oral contraceptive and hormone therapy agent.
Good day, everyone. Today I will be discussing the structure-activity relationship or SAR of two important steroid hormones, estradiol, an estrogen, and aldosterone, a mineral corticoid. SAR help us understand how the structure of these molecules affects their biological activity and interactions with receptors. Estradiol is a major female sex hormone responsible for reproductive health and secondary sexual characteristics. Its activity largely depends on the presence of a phenolic hydroxyl group at the C3 position, which is essential for binding the estrogen receptor. Another important feature is the 17B hydroxyl group, which is necessary for activating the receptor and maintaining its potency. When modifications are made to estradiol structure, they can influence its activity. For example, converting the 17B hydroxy group into a ketone, as in estrone, reduces potency. However, adding an ethynyl group at C17 like ethynyl estradiol increases its stability and bioavailability, making it more effective in oral contraceptives. Additionally, ester derivatives such as estriol valerate improve absorption and prolong the duration of action, making them useful in hormone replacement therapy. Now let's move on onto aldosterone, a vital mineral corticoid that regulates sodium and water balance in the body. Its C21 hydroxyl group is crucial for mineral corticoid activity as it allows for interaction with the receptor and promotes sodium retention. The C18 aldehyde group is also essential for receptor binding and distinguishes aldosterone from other corticoids like cortisol. Modifications to this structure can significantly affect its function. For instance, introducing a fluorine atom at C9, as seen in fluorocortisone, enhances its potency and mineralocorticoid effects. On the other hand, reducing the C18 aldehyde group lowers mineralocorticoid activity, making the compound less effective in sodium retention. These structural relationships are crucial in drug design, helping scientists develop more effective medications with fewer side effects. In conclusion, both estradiol and aldosterone have specific structural features that determine their activity. Understanding these SAR principles allow us to modify these hormones for medical applications such as hormone therapy and managing conditions like Addison disease or estrogen deficiency. Thank you.
Good day, Mom. Today I will be discussing the structure activity relationship or SAR or SAR of hydrocortisol, also known as cortisol. It's a crucial glucocorticoid that plays a major role in reducing inflammation and modulating immune responses. So, understanding its SAR or SAR helps us design better drug with enhanced therapeutic effect and fewer side effects. Let's start with the core structure. So, hydrocortisone has a steroid backbone called the cyclopentanophorhydrophentrine ring, made up of 17 carbon atoms. Several functional groups attached to this backbone are responsible for its biological activity. There are K functional groups that includes the 3 keto group and 4 double band and the 11 beta hydroxyl group and also the 17 alpha hydroxyl group and lastly the 20 keto and 21 hydroxyl groups. So the 3 keto group at carbon 3, which is essential for binding to glucocorticoid receptors, and 4 double band between carbon 4 and 5, which enhances receptor interaction, and the 11 beta hydroxyl group at carbon 11, which is crucial for activating the glucocorticoid receptors. So the 17 alpha hydroxyl group at carbon 17, it boosting glucocorticoid potency. And finally, the 20 keto and 21 hydroxyl groups contribute to mineralocorticoid activity, which affects fluid balance in our body. Now, let's dive into the structural activity relationship of hydrocortisone. This explains how each part of this molecule affects its activity. So the 3-keto group is critical because it allows hydrocortisone to bind effectively to the glucocorticoid receptor. So without it, the drug would lose its function. And the 4 double band between C4 and C5 strengthens this binding and is necessary for anti-inflammatory effect, while the 11 beta hydroxyl group is also essential for activating the receptor. Without it, the drug would have significantly reduced potency. Introducing a double band between C1 and C2 increases the anti-inflammatory effect as seen in prednisolone, a hydrocortisone analog. And fluorination at position like C6 and C9 can also boost the drug potency, which we observe in the drug like fludrocortisone. And 17 alpha hydroxyl group at C17 enhances glucocorticoid activity, while adding methyl or fluoro groups at C16 reduces mineralocorticoid side effects and increases potency, as seen in dexamethasone. And lastly, the 20 keto and 21 hydroxyl groups impact mineralocorticoid activity, influencing sodium retention. Modifying this group can help minimize unwanted side effects. So to summarize the SAR or the structural activity relationship of hydrocortisone, there are a functional activity overview. We have the glucocorticoid activity and mineralocorticoid activity and also the anti-inflammatory potency. So, Anita sa glucocorticoid activity. So, is primarily driven by the 11 beta hydroxyl and the 17 alpha hydroxyl and also the 4 double band, while ang mineralocorticoid activity is influenced by the 20 keto and 21 hydroxyl groups affecting fluid balance and sodium retention. And while the anti-inflammatory potency is enhanced by modifications like fluorination at C6, C9, and adding double bands at C1 to C2. Adjustments at C16 help improve selectivity by reducing side effects. So, in my conclusion in understanding the SAR or the structural activity relationship of hydrocortisone has helped in developing more potent and selective drugs like prednisolone and dexamethasone, which provide a stronger inflammatory effect with fewer side effects.
Prednisone. It is a synthetic anti-inflammatory glucocorticoid derived from cortisone. It is biologically inert and converted to a prednisolone in the liver. It works by decreasing inflammation, slowing down overactive immune system, or replacing cortisol normally made in the body. Cortisol is a hormone that plays an important role in how the body responds to stress, illness, and injury. It belongs to a group of medication called steroids. Prednisone is a corticosteroid used to treat inflammation or immune-mediated reactions and to treat endocrine or neoplastic diseases. The mode of action of prednisone. The prednisone decreases inflammation via suppression of the migration of polymorphonuclear leukocytes and reversing increased capillary permeability. It also suppresses the immune system by reducing the activity and the volume of the immune system. The anti-neoplastic effects may correlate with the inhibition of glucose transport, phosphorylation, or induction of cell death in immature lymphocytes. It may have anti-emetic effects by blocking the cerebral innervation
of the emetic center via inhibition of prostaglandin. SAR, or structural activity relationship of the prednisone. The steroid nucleus. The basic steroid ring structure is essential for receptor binding and activity. The 11-beta hydroxyl group. This hydroxyl group is considered the most critical feature for glucocorticoid activity as it directly interacts with the receptor. The C3 keto group. The presence of a ketone at the C3 position is also important for receptor binding and activity. The 17th alpha hydroxyl group. This group contributes to the overall potency of the steroid. The 21 hydroxyl group. While not as crucial as the 11 beta hydroxyl, the 21 hydroxyl group also plays a role in receptor interaction. Prednisone overview. Its chemical nature. Prednisone is a corticosteroid, meaning it is a steroid hormone with a specific chemical structure, including a ketone group at the 11 positions. For the conversions to active form, when ingested, prednisone is converted by the liver into prednisolone, which has a hydroxyl group at the 11th position instead of the ketone group. Now, for the importance of hydroxyl group, the hydroxyl group in prednisolone enables it to bind more effectively to glucocorticoid receptor, leading to its anti-inflammatory and immunosuppressive effect.